So, and I wanted to start with these graphs that you have seen already, probably, I suppose, that this is from the latest IPCC report. And I want to start with that, not because it's something you don't know. You probably, I hope you do, that the climate is changing. And but I wanted to make it clear again how serious it is and what we are talking about. Here's a map of Europe for different variables, and the colors are indicating the change as compared to the situation now. So I copied the three graphs about heat, precipitation, and drought. And you can see there the two scenarios, the plus 1.5 degree and the plus 3 degrees. But basically, especially in our areas, Belgium, Germany, and so on, you see that there is color, meaning that there will be change. And in most of these graphs, this is a change towards more heat, more extreme precipitation, and more droughts. So that's something that we will have to take into account. And if you look at this graph where they try to reclassify the climate, the future climate, according to our Geiger climate classifications, you can see that we are basically moving into different climate zones. And I think that's quite important for the topic of today. That's why I wanted to show it again. So we're really moving to a different to different situations. We have to do everything to stop that as much as possible, but we'll also have to adapt, right? And that means for us here in Belgium, and I think for large parts of Germany, that's also the case, that we both will have more extreme rainfall events, where if you then think about agriculture, that will mean more frequent flooding, also erosion problems might aggravate. There will be issues of anoxia in fields. But it also means more frequent droughts, water stress, salinization problems that are also occurring in the coastal areas in Belgium, for example. So yeah, as you all know, these things are quite important to think about if you're in the agricultural sector, and farmers are already feeling it in their yields, and insurances too, because I don't know how it's done in Germany, but in Belgium, the yield loss is covered by an insurance which is partly covered by the government also, and there every year in the past 4 or 5 dry years has been covered by these insurances. So I didn't want to make you feel bad this morning, so I hope you don't have this climate dip now that sometimes you can read about in the papers. But what I do hope is that we realize that in agriculture, we'll have to deal with that. And so what I'm talking about today is the phenotyping. And now I'm coming to the topic, because the good news is that we are creative beings, and I really do think that we still have some ways in agriculture also to adapt to these changes. As I said, of course, the first thing is also to work on mitigation, but we'll also have to adapt. And breeding is one of the key weapons we would have, I would say. It's something that we have been doing for decades and even centuries, right? We are selecting as human beings always the best agricultural crops. And it's something that we have been doing and that we will have to keep on doing. And long-term breeding is basically the way to go. Why? Because there is not one single condition in which you can select a crop that will then work 100% for the next year. Here you see an example of this could be a normal year for a certain crop, but then if you would select the best performing variety in this year, if then the next year, for example, at the end of the growing season, there is a drought, it might be that not the same varieties are performing well. Or if you then continue and you might have a year where it's rather dry in the beginning of the season, just after emergence of the crops, or it's an exceptionally dry year, we still want to be able to have a robust crop that can perform in most of the conditions of a certain climatic zone up to the standards that we need. So it's not as simple as just selecting in one trial the crop that is dealing best with a certain condition, but it's selecting actually a crop that can deal with the variability also of meteorological conditions that we can expect now and in the future. And that's basically what we are talking about now. The only difference might be now, as compared to 20 years ago or earlier, that the changes that we see now are much faster than than what we saw in the past, and so adaptation becomes even more important. So yeah, so then the question is, if we do this kind of trials or we work on long-term breeding, we still need to know what to look at, right? So this is a picture of a typical breeding trial of grass at ILVO, and you can see all these different varieties. There was a drought then, or in this case, we imposed a drought with shelters. And the question is, okay, how can you see which crop is performing well, which crop should stay in the pipeline or in the breeding process, and which one doesn't? That's basically the field of phenotyping, right? We are going to select certain traits, that's what we call them, to look at, and those, and we assume that those can represent the performance of the crop and also bring us closer to understanding why this crop is behaving like that. So in that way, we can also maybe link genetics with what we see when the crop is growing. And in phenotyping, you have many different practices, from very controlled conditions in the lab, greenhouse, up till the field. And that's actually what I know, that also in PhenoRob, I don't know who is actually today in our virtual room, but I know that a lot of researchers in PhenoRob are working in one or several of these scales and techniques, let's say. So I'm also keen to hear from you in the workshop, what are the recent advances in these? Basically at ILVO, we are interested in the bigger scale, let's say, the scale where we work already in the field conditions. And let me explain why that is. Because we know that there is, a crop reacts to its environment and is not only determined by its genes. How a crop will develop based on its genetic material, you can test in the greenhouse. You can already impose some treatments or change the environment in a greenhouse and then see how a plant reacts. But the problem is that we know, we've seen in scientific research, that there's not necessarily always a super correlation between how such a plant reacts in the lab, for example, or in the greenhouse to a drought treatment, and how it reacts then in a real field and production environment. And so I think it's very important that we do not forget this intermediate scale before seeds are going really to the farmer, of course. That's this testing in the field. And of course, that brings some challenges, because it's not always possible to have the high degree of technological, high degree of technology that we have in the greenhouse, also in the field. And that's also a challenge that a few of you are working on. There are some robots that are being developed or already used actually commercially, and other techniques. Just to give you some examples, something that's already quite standard also with breeding companies is working with remote sensing and with drone data. Why? Because this allows to still be quite detailed, still gather quite detailed and information about the crops in the field. You can still test a lot of crops because you can image or you can take pictures of many of those varieties at the same time. Here you see an example of soybean, where with which we have a lot of experience in ILVO, because we just have created a spin-off also that actually sells soybean seeds and is doing the breeding process from now on. Here you see an example where we assessed the plant height, for example, as a specific trait of the plants with drones. And the nice thing is that you cannot only do that once, but you can do that several times and then establish growing curves and look not only at static traits of plants, but also at dynamic ones, right? So here you can see some examples of what it can give, and that's something that's there's still a lot of advances to make, or the ways to use drone cameras and different sensors and so on are still progressing, but this is also something that's already commercially applied. Another example is to look here at grass, I think this was, yes. And you can see the temperature that is assessed with a thermal camera on the drone. So basically, plants that are closing stomata heat up more than those who aren't, and then you can see differences between how plants react to heat and drought stress. So this is more to give you an overview of what's already more common and where we are also still doing research at ILVO. So But the question here today is it only important to look at what we see above the ground, or should a breeder also start to have more information of what's below? And I think we all know the answer. For me, the answer is, I think there's, and I think that's also something that's clear in the literature, not also for me, that we need more information also about what happens below the ground. That's where the roots are, where nutrients are, where also interactions with microbial life and other soil components is happening. So to really understand how a plant is dealing with its environment and how different varieties are differing in that interaction, we also need below ground phenotyping in the field. And then that's the difference with what we already can do in the lab. We can already do quite a lot in high throughput phenotyping installations on root system characterization and so on in controlled conditions. But in the field, it remains a challenge. And why is that? It's of course because you cannot easily look into the ground. You cannot see what's in there. And so the challenge is, how can we study those roots in the field without completely destroying or altering the environment while doing it, right? It's an old question, but it's particularly relevant here in this phenotyping question. And the need is really there. This also, I can also say that we've been already approached by several companies in the breeding industry that are really looking into how can we get more reliable information on roots and how roots are interacting with the soil and with their environment in the field. So right now there's already some literature on that and some methods that are available. Here you can see 3 of them that are mentioned in this paper by Wasson in Trends in Plant Science. It's just to illustrate it, but it's going from techniques that are quite manual and still invasive, where you have to use a spade. It's called shovolomics to look at the root system at the top. Yeah, it could also be in other ways, like with samples, scoring, things like that. So the basic thing is that you still have to do invasive sampling, but in the sample then you can do imaging, and there are several other methods that you can actually, even in automated pipelines, use to study that root zone, let's say. The problem there is that you still have to disturb your field. And if we're talking about field facilities with shelters and so on that are remaining at the same place, that can be quite problematic actually. Yeah? If you have to every year take a lot of spades of soil and then you repeat that 15 years, at the end, your field will not be the same anymore. And that's particularly important for plant scientists. Before I'm a soil scientist, so I'm used to taking samples and digging a lot, making profile pits. And sometimes people almost get a panic attack when I'm trying to say that I want to dig a bit in the soil where they are doing the breeding experiments. So this is really an issue actually. Another thing that you're also working on, for example, in the facility, I think it's in Zilhausen, you have this huge and great mini rhizotron facility. It's very interesting. It gives also, not information on the whole root system, but on particular areas where the tube is. And there's always also discussion with this kind of methods that by putting the tube, you alter the environment, of course, in which you put the tube. So it's very important to work then on long-term installations where this disturbance can be minimized over time, let's say. And as I did mini rhizotron research myself during my PhD, I can say that it's still a huge work. All these images and camera work, it's still very time consuming. And then the third like range of techniques that comes up there in the literature, and that's basically also where my research is often situated at the moment, is to look at how can we do non-invasive sampling of the below ground plant parts, and that can be done with geophysical techniques. They all have their flaws, of course. Here on the picture you can see EMI. I don't know in how far everybody knows geophysical techniques here, so let me just briefly explain what EMI is. It stands for electromagnetic induction, and it's a technique where we actually, to say it very simply, we work with a magnetic field. So we turn on a magnetic field, which is time varying. This induces an electrical field in the soil. Due to several different conductivities in the soil, we can create then in the soil, secondary magnetic fields are created. And because we then have a sensor that can sense the magnetic fields, we can see this reaction together with the initial field, and then we can induce something about the conductivity of the soil by sensing this secondary magnetic field. Why is that so important? Well, I'm showing this because actually one of the first papers that started to really show that geophysics could not only be interesting for scientists studying soil-plant interactions or looking at eco-hydrological questions, but the first scientist who applied it to really a more breeding environment was Wally et al. In the group of Lancaster. And there they use specifically this EMI technique to look at differences in wheat varieties and how the below the ground, how the electrical conductivity below the varieties varied with depth. Here you can see an example of the famous paper that is often shown in presentations on this kind of topics. You see the different dates where they did measurements with this EMI technique, and you basically see over time that the soil is drying out. The soil becomes more resistive. When water is disappearing, the electrical conductivity is reduced, so the soil is more resistive. And basically there you can see that the idea of this, this first attempt was, are these techniques good enough to actually see differences in uptake of water by these different varieties? It's an early attempt. It's not yet a very quantitative attempt, and that's also partly related to the choice of the EMI, in my opinion. But it was, it was inspiring to see that this was maybe something that was worth exploring. And after that, and also even before already, there were several people working on this topic. On EMI, for example, Guillaume Blanchy, that was coming from the same group, but also with ERT in my group, but also in other groups, people started to work on, okay, even, I did not mention, but I put this paper here of some colleagues of you, Maximilian Weigand, Valentin Michels, and Andreas Kemna, are working already since I'm doing also since I was doing my PhD, they're actually also working in this field. So basically there's a lot of potential, and many people believe that, that this potential is there, but it was still lacking, I think, up till now, some application which was scalable, let's say, not only a few transacts that a researcher has to follow up on, which is very time intensive and also very, which requires a lot of expertise. And so the challenge that I wanted to take on at ILVO was to try to build a facility that would use these techniques in as simple as possible way, let's say, so that also it could be run automatically. It would give real-time data, and we could then use it to convert it to breeding traits, and to send that together with all the other traits in a breeding experiment to the researchers or the breeders that were interested in them. So we were able, luckily, to get funding from the Flemish government for an open research infrastructure, which is, as it says in the name, open, so it can be used by any research group internationally also. So I'm happy to work with you in the future if this could be of interest also for PhenoRob colleagues. And so in this facility, we wanted to combine the above ground phenotyping techniques that we were already using a lot, the drone, but also the classical breeding observations that we are still doing. We have quite a lot of expert colleagues on different crops that can also assess traits with very classical techniques. But then we wanted to add the below ground phenotyping part to that. So that was my addition to this project. And we wanted to include also the possibility to not only assess the current environmental conditions, but also to do trials where we could induce drought or induce rainfall in the way that we wanted to simulate a certain environment, right? So this facility is actually now being built and should be ready in, I think, about 1 month. Everything should be ready. Part of the ERT is already in the ground. The shelters are almost there. And the drones are waiting to be deployed over there. So how is this facility working? Just to give you an example, here, the drawing is, of course, not at scale, but what you can see there is that there are several shelters, and each shelter is serving 3 fields. One field has the size of 30 meters on 10 meters. And in one of those fields, you can do a breeding trial with, depending on the trial you want to do, plots or single plants, combinations of, 10 to 100 of varieties, depending on how you lay out the field. And why do we have each time 3 fields and not just one? Well, because we also respect an agricultural rotation. So a field is only used for a trial every once in 3 years, actually. So that's, we don't have 5 years in a row, for example, wheat on a field, and then also the subsequent problems with diseases and so on. Plus, it's also necessary to homogenize the soil again after a trial with little plots and things like that. So back to the below ground then, now that you have an idea what this looks like. Let me also look at my time. I have to finish almost. So this electrical resistivity tomography is a technique where we use electrodes instead of coils to there you induce the magnetic field with the EMI. With ERT, we just use electrodes directly in the soil to insert currents. We measure the voltage at other electrodes, and that gives us an idea again of the electrical resistivity of the soil. And by combining many combinations of those injection and measurement electrodes, we can then retrieve a distribution of electrical resistivity. That's already interesting, but then what makes it really interesting is when you combine this or when you repeat this in time. So repeat this in the beginning of the growing season, and then every day or several times a day, the same measurement at exactly the same place. And then you will see differences between measurements, and those differences are exactly what we are looking for, because in an environment, for example, in drought, these are related mainly to the root water uptake of the plant. I see that I don't have a lot of time to go into the details, but I just wanted to say that we did a synthetic experiment to determine how we best do the layout. And I even talked also with was it Valant? I think it was actually Maximilian about what were their experiences also with the EIT experiment. And so in the end, after the synthetic study, which I will skip now, I can give you more information later, we went for a layout where we had a surface cable and a cable buried at 50 centimeters depth, so just below the plowing depth, or a bit below the plowing depth, so we were sure that it could stay in place forever and that the plow could just move over the field without any issues. So to go directly towards the important points, so we did a synthetic experiment where we tested those different layouts, and we also imposed different root water uptake depths, or also root growth, different rooting systems, let's say, how deep they go. And then we compared how well we could retrieve those virtual root water uptake profiles from then virtual ERT measurements. So this is not in reality, this is all using soil, water, and geophysical models, let's say. But basically, like that, we could test, A, are we able to retrieve, for example, a maximum root water uptake depth, and B, in which soils is that possible? Because sometimes if the soils are too conductive, for example, that could be problematic. And so these curves were obtained. We could see that actually in different soils, that's what you see in this graph here. You see the root mean square error between what we know was there in the model initially, the root water uptake curve, and the one that we could retrieve from virtual ERT measurements. The brighter the color, the more yellow the color, the bigger the error. And so you can basically see that just the surface transect was not enough. Those are the different lines of this graph. But if you use buried cables or boreholes, you can get a better RMSE. And what you also see is that it does matter which soil type you have. So that's maybe also something to keep in mind also towards the scalability. If you would want to repeat this in a clay soil, for example, you can see that already you can expect bigger problems or bigger errors, let's say, between the ERT and the original profile. But basically, we saw that with the buried cables, it worked. And so to come to the end, the main target of this ERT here is mainly to be able to, for example, retrieve the depth of maximum root order uptake, so which would be the peak of this curve of this profile, or also to see what's the deepest, where are the deepest roots, or up till which level water can be taken up, so the deepest depth of root order uptake, and maybe even some 2D patterns. This should be possible in the tomography, but we will see how easy it is to distinguish between varieties if they are close to each other in a field, of course. So this is my end message. I think it's not only about measuring the below ground properties, but it's also about then knowing what to do with them. Just knowing that there is a certain root order uptake depth and then linking that to performance is probably not enough for breeding. So I think our next challenge will be, after we get all this data and we just look at them statistically, will also be to combine them in a real data processing pipeline where we use mechanistic models, but maybe also machine learning or deep learning algorithms to basically link the below ground traits to above ground traits also, so that you know when you see something at the top, when it might be related to what's happening below, and also to explain why, what are the processes behind a certain performance. To conclude, in this installation, we are trying to link below ground phenotyping with what we see at the surface. And I think that's really one of the next big things for the phenotyping, for the breeding companies, to see in the field quickly which traits matter for, to get a performant crop. Geophysics has really a role to play here. I really believe that our techniques are very valuable, and we have to keep developing them and make them more performant to serve this community. And I think it's also it's also important to state again that this is an open research infrastructure. So I'm looking forward to meeting you there and to work together, because I think there's many possible experiments and layouts we could try there. That's it. I just wanted to say I also wrote a book about water and agriculture and other fields in general, and it's now translated in Dutch, in German also. So if you would like to have a closer look at what my research and my interests are about, you can also have a look there. It's made for children. And there is a job in our installation here for plant phenotyping. So I also put the QR code here for people that think, okay, my PhD is almost finished and I would like to go for a next job. Have a look, please.